Okay, students. So, in continuation to the previous lecture on transport across membrane, I'll talk to you about transport proteins. Right now, we have already discussed the significance of these membrane proteins, like these transport proteins, in both passive and active transport, right? Today we learn about what are these transport proteins, and we'll talk of their role in active transport and the type of active transport. So transport proteins have much in common with enzymes. Enzymes are proteins, and they can have specific binding sites for the solute. Right, once we had discussed about the different type of membrane proteins. I told you these membrane proteins can be either channel proteins or carrier proteins, right? When we talk of channel proteins, they just make a passage to allow the movement of molecules across them. But when it comes to carriers or specific transporters, right, they have binding sites for these specific, all these proteins being specific, they have specific sites on which the solute, which they are going to carry along with them or transport the solute either from cell interior to exterior or vice versa, they have a binding site for that particular solute. So transport proteins can become saturated when they are moving passengers as fast as they can. Now, this is very important to understand. I mean, a protein, a transport protein, which is carrying a solute from one side of the membrane to another. Right, it is going to have a particular capacity on the total number of solute particles. Say, for example, it can carry, it can be one, two, three, depending upon how many binding sites it has for that particular solute. So it can be assumed that at a particular moment or at a particular when those sites are occupied by the maximum number of solutes that can be carried by that particular protein molecule become saturated. Now, if we take a look at it in terms of transport of that particular solute across membrane, then during that time frame, unless and until it has carried that maximum number of solutes and transported from one side to another, no more molecule can exist there. So it is true that these proteins can become saturated also. Transport proteins can be inhibited by molecules that resemble the normal substrate. See, when we talk of enzymes, we always know there are inhibitors, right, that can block enzyme activity, that can hinder enzyme activity by disturbing the active site, blocking it, or decreasing its efficiency. In the same way, many of these transporters are inhibited by substrates or molecules that resemble its specific solute which it carries, right? If that particular, substrate goes and binds there, it will lead to the saturation of those sites, the binding sites of that particular solute, and it will no longer be able to carry that specific solute across the membrane, right? So I hope you are clear with all these points. Okay, let's move ahead. Then comes the channels. Channel, as you know, it means a passage, as you can see here, right? They do not have, a carrying capacity. They cannot change conformation. I'll just, uh, I'll just come back here. Yeah, if we talk of channels, they provide corridors which allow a specific molecule or ion to cross the membrane. Right, they just have a passage that allows the flow of molecules both actively or passively, right, across them. There are different channel proteins, and again, like other membrane proteins, they are specific in what molecules they are going to allow to be transported to them. Of course, it has been seen that in comparison to the carrier proteins, right, they which bind to a particular solute, then at say at one side of the membrane, carry it and move to the other side of the membrane and transport it. These channels they allow fast transport in comparison to them. Water channel aquaporins, they are called as aquaporins, make possible massive amounts of diffusion. This is one example here. I just want to quote that. We have talked of easily through the brain also. But for fast transport of water molecules, right, for fast diffusion of water, some membranes have these proteins called aquaporins that allow the movement of water or diffusion of water through them. Now, these channels again, they are of two different types. They can be either gated or non-gated. Non-gated channels normally are allowed. They allow the movement of molecules passively through them according to their concentration gradients, right, from a region of high to low concentration gradient. But if we talk of the gated channels means these passages are not always open, right? They have a gate, that is an opening and a closing system, or in terms of protein, we'll say a change in conformation, which allows the movement of molecules under specific conditions only. As you can see here in this figure, that this channel proteins, this is a gated channel. In response to a certain stimulus, it is allowing the movement of molecules through it. It has two openings, one towards the extracellular side and one towards the intracellular side. So unless and until this extracellular side is open and allowing the movement of molecules across it, is not possible. So gated channels open or close depending on the presence or absence of a physical or chemical stimulus. For example, neurotransmitters, they bind to specific gated channels. On receiving the neuron, these channels open. On receiving some stimulus on the brain, say an action potential is generated and nerve impulse is conducted. Once they receive that impulse, right, that neurotransmitter is released. It might go and bind to say some channel protein on the target organ also, and then it opens. Gated channels, they require some stimulus. For opening means in simple terms for allowing the movement of molecules across them, and this opening and closing is a change in conformation of the protein, simply meaning that in the absence of a stimulus, the conformation of the protein is such that the movement of that particular solute is not possible through them. But when a stimulus is present there, it leads to a change in conformation of the protein so that it is now possible to allow the movement of that solute across it. Okay. So, for example, many neurotransmitters allow sodium ions into a nerve cell, and when the neurotransmitters are not present, these channels are closed, that is, they do not allow the movement across them, right? So, we have some non gated channel proteins and then specific gated channel proteins that open and close and allow the movement of molecules across them, solutes across them in response to a specific stimulus only. So, are you clear with this difference? Okay, let's move ahead. Okay, we have talked of passive transport in the previous class, right? Now, today we will be talking about active transport, and we have already discussed on the major difference between active and passive transport. So, the first and the foremost difference that applies to both foremost difference that applies to active transport is that it requires ATP and energy. And the second thing is that it moves solutes against their concentration gradient in comparison to the passive transport, which allows the movement of molecules down their concentration gradient, that is from a region of high to low concentration. But suppose if the membrane has to transport a solute against concentration gradient, that is from a side where the concentration is low to a side where the concentration is high. So there the kinetic theory will not apply, right? There is I mean this movement thing in the form of energy that it can be transported from one side to another, and that too from a side where the concentration is low to where the concentration is high. Okay, so they move solutes against their concentration gradient, that is from the side where they are less concentrated to where they are more concentrated. These are vital for a cell to keep up its internal concentration of small molecules that would normally diffuse away. Now, sometimes what happens is that there are certain ions, say for example, inside the cell, right? Their intracellular concentration is high as compared to their extracellular concentration. And due to that, they have a tendency to diffuse to the other side, right? But in this case, I mean, a drop in their concentration, although it's high, but if some of these molecules they also diffuse away, it leads to a number of disturbances inside the cell. It can stop some cellular process, right? So, I mean, the diffusion is not so intense that the concentration to the extracellular side is high, but still, even if some molecules they diffuse away, the intracellular concentration falls. So the cell cannot wait for passive transport to occur to retain that particular concentration inside. So in that case, this active transport helps to retain that intracellular concentration by transporting some of those molecules against their concentration gradient, and thus helps the cell maintain the internal concentration of its vital constituents to a optimized level. Again, this is done by specific proteins which are embedded in the membrane. Any transport across membrane, right? Specialized transport and normally large molecules, non-polar molecules, they always move through these embedded transmembrane proteins. This you should remember. Then active transport is categorized into two types. One is primary active transport and the other is secondary active transport. So we'll take up both these type of transports one by one. Now, if we talk of primary active transport, okay, it is also called direct active transport, as it directly transport molecules across a membrane. We'll try to understand this with the example of the most common pump that is known, that is the sodium potassium pump, which helps to maintain the cell potential, that is the potentials within cell across membrane, by maintaining the concentration of sodium and potassium ions both intracellularly and extracellularly. So, sodium potassium pump maintain the gradients of sodium and potassium ions across the membrane. Typically, animal cells have higher concentrations of potassium and lower concentrations of sodium inside the cell. So the sodium potassium pump uses the energy of 1 ATP to pump 3 sodium ions out and 2 potassium ions in against the gradient. Okay. Now we'll see how this pump works with the help of this diagram. Okay, is this diagram visible to all of you? Okay, so you can see the different steps are mentioned here. Okay, so in the first step, now I hope you recognize this is the plasma membrane, right? And this is the pump. At each and every step, it is shown the different conformations and the different sites being occupied by different ions, that is sodium and potassium, how they bind and how they are released, and how ATP is consumed. So we'll start with the first step, right? This is the extracellular fluid and this is the cytoplasm. So binding of cytoplasmic sodiums to the protein stimulates phosphorylation by ATP. So whenever this pump has to transport, I mean with consumption of each and every ATP, we have read here that it uses energy of 1 ATP to pump 3 sodium ions out and 2 sodium ions in, 2 potassium ions in against their gradient. Okay, so now, whenever the cells intracellular concentration of sodium rises, right, above the normal levels, then in that case, that excess of sodium is allowed to bind to the sodium potassium pump. Okay, it has three binding sites, right? But in order to transport these sodium ions outside the cell, it has to use or it utilizes the energy of 1 ATP. Okay. Upon ATP hydrolysis, you'll see this free phosphate is bound to this protein. Okay, this protein is phosphorylated, and upon phosphorylation, the conformation of this protein changes such that it causes the protein to change the conformation, and the conformational change expels sodium to the outside and also allows the binding of extracellular potassium. So this change in conformation actually opens this gate towards the extracellular side, or allows such changes in conformation that sodium is no longer able to bind here, right? This leads to the release of these bound sodium ions outside the cell. Now, this conformational change also allows the binding sites for potassium ions to be exposed such that two potassium ions, I mean, as soon as these three sodium ions are released, potassium binding triggers release of phosphate group. As soon as these two potassium ions they bind, this phosphate is released. I mean, again, the protein conformation changes. Now, you know, whenever this protein is not phosphorylated, in its original state, it has a tendency to open up towards the cell interior. So, this only happens. Binding of two potassiums releases this phosphate from the protein. The protein is dephosphorylated, leading to the opening of the protein towards the extracellular side, and thus releasing two potassium ions inside, right? Now again, if the intracellular sodium concentration is still high, potassium is released, and now again it is free to bind to three sodium ions, because again opening of this channel towards the intracellular side and its dephosphorylated state is the most optimum condition for binding of sodium. So in this way, these cells they maintain their intracellular concentration of sodium and potassium by transporting these ions against their concentration gradient through active transport. I hope this example makes it more clear to you that why this ATP consumption and how this ATP consumption allow the changes in conformation of this membrane protein in terms of its phosphorylation and dephosphorylation that helps the cell transport sodium and potassium ions. Okay, please go through it, and if you have any questions you can post here. If you are comfortable, please type OK so that I can move ahead. Everyone, please, I'm waiting for a response. Okay, let's move ahead. Now we'll talk of the second type of active transport, that is called secondary active transport. It is also known as co-transport, as it uses energy to transport molecules across a membrane. It comes under active transport. However, in contrast to the primary active transport, here there is no direct coupling of ATP. Instead, the electrochemical potential difference created by pumping ions out of the cell is used here. It is instrumental here. I mean, once suppose a protein, a channel protein is allowing the transport of a molecule across it, right? So due to that transport, the electrochemical potential difference that is generated, that energy is actually utilized to co-transport a molecule with it. So that is why it is called secondary active transport, because it is not directly using ATP, or it is not directly coupling to ATP to transport a solute molecule across the membrane. So the two main forms of this secondary active transport that are known are antipode and sympode. Now, antipode, as the name defines, that is, anti means opposite. So, in antipode, two species of ion or solutes are pumped in opposite directions across a membrane. I mean, if one molecule is being transported, or one solute is being transported from cell exterior to cell interior, then the other will be transported from cell exterior to cell interior. It is always going to be in the opposite direction. That is why it is called anti-transport. One of the species is allowed to flow from high to low concentration, which yields the entropic energy to drive the transport of the other solute from a low concentration region to a high one. The most common example of antipode is the sodium calcium exchanger, also known as the sodium calcium antiporter, which allows three sodium ions into the cell to transport one calcium ion outside. So as soon as three sodium ions are pumped inside the cell, one calcium is transported outside. But please do not confuse, all these transporters are different, right? I mean, you'll have a different sodium potassium transporter, then we'll have a different sodium calcium exchanger. All these are membrane proteins that you have seen. Once we have talked of the mosaic fashion of proteins being present in the membrane, it is all these proteins, different type of proteins, that are required for different types of transport across membrane. Then comes sympode. Sim means same. So sympode uses downhill movement of one solute species from high to low concentration to move another molecule uphill from low to high concentration against its electrochemical gradient. However, the direction of transport will remain the same. For example, the glucose symporter SGLT1 co-transports one glucose or one galactose molecule into the cell for every two sodium ions it imports into the cell. So you will see the direction of transport is always same. However, the concentration gradient is different, right? One molecule is moving passively while the other is utilizing that electrochemical gradient that is generated due to that passive movement to actively transport another molecule, but in the same direction. Okay, so are you clear with sympode and antipode? Okay, fine. Then, when we talk of transport across membrane, we must always talk of these two terms, that is exocytosis and endocytosis. Okay, these are ways of getting large molecules in and out of the cell. And we always talk of two terms, that is phagocytosis, which is also known as cell eating and involves solids, whereas spinocytosis is cell drinking and involves liquids. Okay, so these are again two important processes that allow the movement of molecules from or some, you can say, some larger molecules to move from outside to inside or inside to outside. Exo means to exit, so something which is transported outside the cell follows exocytosis, and endo means something is moving inside the cell. We have already discussed this in detail, but since we are talking of transport across membrane, we must talk of these two terms. Now, here you can see the electron micrograph picture of phagocytosis in a bacterium, where you can see a gray colored structure here resembling the membrane. This is the extracellular fluid. And you can see these pseudopodia or finger-like projections which are formed in the by the you can say projections of the membrane. They carry this food, or you may say some other particle, inside the cell. They form a membranous vesicle that is then transported inside the cell and utilized. So this is phagocytosis, is a type of cytosis. Then you can see another example where it's given to be pinocytosis, where you'll see instead of large solid type of particles, some fluid particles can also move inside the cell through these vesicles which can carry these liquid particles inside the cell, and this is known as pinocytosis. Then there comes a specific type of endocytosis which is called receptor mediated endocytosis. It is very specific because any molecule that is endocytosed or moved inside the cell, right, it is allowed only through binding of specific receptors. These structures which are present here that are specific to the solute, they are going to bind, and these are actually present on the surface, on the extracellular surface of the membrane. They bind to their specific solutes or molecules they are going to carry. Once these receptors are occupied, this receptor ligand interaction is done, that part of the membrane invaginates and coated vesicles transport molecules inside the cell. We'll talk about its detailed mechanism once we'll talk of vesicular transport in eukaryotic cells. But for this moment, you should know that endocytosis can be specific and non-specific, right? So if we talk of receptor-mediated endocytosis, highly specific because it is going to of molecules, of only those molecules rather I should say, that are going to bind to these receptors. So this is all a introduction to transport across membrane. What are the different processes? What are the fundamental things that you should know? What are the different types of transport that different membranes can perform? Thank you so much, and we will talk about a new topic in the next class.